Section 6.2, the process of meiosis. Write the notes for the next two slides on the handout you were given in class. Meiosis is a form of nuclear division that creates 4 haploid cells from 1 diploid cell. All of these cells are genetically unique. This process is how sperm and egg cells are created. It occurs in 8 steps with 2 main phases, meiosis 1 and meiosis 2. Pairs of homologous chromosomes separate in meiosis 1. These are homologous chromosomes. Sister chromatids divide in meiosis 2. These would be sister chromatids. Homologous chromosomes are similar in structure and types of genes but are not identical. Sister chromatids are copies of the same chromosome and therefore identical. These are formed during the S phase of the cell cycle when DNA is being synthesized. For this next part, use pages 2, 3, and 4 of the handout you were given in class. Draw the appropriate drawings for the 8 different phases and put any information you need to the left-hand side of the circles. In prophase 1 of meiosis, the chromosomes would begin to condense. Homologous chromosomes would pair up. The nuclear membrane would break down. Spindle fibers would begin to form, and crossing over would occur during this phase. So if this is our cell, this is a nuclear membrane starting to break down. Spindle fibers would start to form, and our homologous chromosomes would pair up. Refer to figure 6.2.3 on page 333 of the textbook. Crossing over occurs during prophase 1 of meiosis, so you're going to write this information on the back of the box for the prophase 1 slide, which is on page 2 of your handout. Crossing over occurs when homologous chromosomes exchange genetic material. Genetic material is said to be recombined or forms recombinant DNA, adding to genetic diversity or creating genetic diversity for that organism. So these would be our homologous chromosomes. And as you can see, these 2 arms have literally crossed over, okay? And they're going to break off and reattach. So you're getting part of this one that reattaches here, and this one that reattaches here, forming 2 recombinant chromatids, where this and this exchange, and these 2 non-recombinant chromatids. Metaphase 1 of meiosis, the homologous chromosomes randomly line up along the equator or along the center of the cell. The random arrangement of both paternal and maternal chromosomes mixes up the combinations, ensuring genetic diversity in the gametes that are being formed. So here's our cell, and we have our chromosomes, homologous chromosomes, lined up on the equator. these guys. And there we go. Metaphase 1. In anaphase 1, the spindle fibers shorten and the homologous pairs of chromosomes are separated and move towards opposite poles. The sister chromatids remain together. So if we're looking at our cell, this cell is going to elongate. It's going to look a bit like that. The sister chromatids are going to be pulled towards the poles, like so. So these are still paired up chromosomes. The spindle fibers have shortened, which pulls them this way. But the sister chromatids are still linked together, so we have these paired up chromatids. In telophase 1, the nuclear membrane forms. In some species, the spindle fibers break down and disassemble, and the cell would undergo cytokinesis, forming 2 new cells. So these are 2 new cells, right? Cell membrane will begin to, or the nuclear membrane begin to reform. We have our chromosomes. There and there. Okay. In humans, you get 23 duplicated chromosomes, some from both parents. The chromosomes could be from either mom or dad, depending on how they lined up during metaphase 1, how they lined up across the equator. In prophase 2, the nuclear membrane breaks down, the spindle fibers form, and our 2 cells are getting ready to divide again. Okay, note that there is no DNA replication between the 2 cell divisions. Between telophase 1 and prophase 2, DNA does not replicate. In metaphase 2, the chromosomes, again, are going to line up across the equator of the cell. Okay, in this case, we're just showing the 1 set of sister chromosomes, right? The spindle fiber has formed all the way from the poles to the chromosomes. Okay, and they're lined up on the equator of the cell, getting ready to divide. In anaphase 2, the spindle fibers shorten and the sister chromatids start getting pulled to the poles of the cell. In telophase 2, the nuclear membrane reforms around the chromosomes in the cell. So we're starting to get our nuclear membrane to reform. And we've got our 1/2 of the sister, or 1/2 of the chromosome, the sister chromatid in each one. And the result that forms here are 4 haploid cells that are genetically different from one another. Okay, go to Google in your browser and Google meiosis animations and watch 1 or 2 or more of these animations of meiosis to get a better idea how to visualize and understand the process of meiosis. Copy the information for the next 3 slides onto the notes handout under number 3 on page 1. Use the back of that first page if you need more room, and if you need to draw anything, please do. Gametogenesis is the production of gametes. It marks the changes that take place in haploid cells formed from meiosis before they are actually functional as sperm and egg cells. Gametogenesis is different between males and females. In spermatogenesis, 4 sperm cells are produced. Sperm becomes streamlined and develop a flagella. This is used to move the cell. It's a whip-like tail that's used for swimming the sperm cell to the egg. Sperm cells are pretty much mostly nucleus with this long tail and some mitochondria to kind of move them along, but they primarily contribute DNA to the developing embryo. So if this is our sperm cell, germ cell, I should say, and it's going to go through the first meiotic division and form 2 cells, which will go through the second meiotic division. Both of these divide again, forming 4 haploid cells. Okay, that's the cell membrane, the 1 chromosome we're putting in there, and the nuclear membrane. And these guys are going to streamline, get a little bit elongated, get this bunch of mitochondria here and this long tail. So then they develop into the sperm cell. Oogenesis, or formation of the egg cell, during meiosis 1 and meiosis 2, one cell gets most of the contents to produce the egg, or ova. The cytoplasm is split unevenly. The smaller cell becomes what's called the polar body. These things don't survive. The eggs, since they have most of the cytoplasm and all this stuff in it, contribute DNA, but also cytoplasm, organelles, nutrients to the developing embryo. So if we have this oocyte, that's going to divide during the first meiotic division, or meiosis 1. Most of the cytoplasm goes into this cell, and very little goes into this cell. It still gets the same amount of DNA. That's the same. I mean, it's different combinations, but it's the same amount of DNA. And then during the second meiotic division, again, this guy divides unevenly, so we get 1 egg cell, which has almost all the nutrients, organelles, and cytoplasm, and these 3 smaller polar bodies, which just dissolve and go away and don't develop into anything. So this would be the egg cell, and these 3 would be polar bodies.